Do you know that nutrition is an important environmental factor that affects your genome? Welcome to ADP for Learning. I am Ashish Katyal. Today, we are deep diving into the topic of nutrition and its effects on our genome. As shown in the figure, nutrients from our food are funneled into a biochemical pathway that extracts methyl groups and then attaches them to our DNA. Unlike behavior or stress, diet is one of the more easily studied, and therefore better understood, environmental factors in epigenetic change. The nutrients we extract from food enter metabolic pathways where they are manipulated, modified, and molded into molecules the body can use. One such pathway is responsible for making methyl groups, which are important epigenetic tags that silence genes. Familiar nutrients like folic acid, B vitamins, and SAMe, S-adenosylmethionine, a popular over-the-counter supplement, are key components of this methyl-making pathway. Diets high in these methyl-donating nutrients can rapidly alter gene expression, especially during early development, when the epigenome is first being established. It is important to note that diet during early development can have long-lasting effects. Your mother's diet during pregnancy and your diet as an infant can affect your epigenome in ways that stick with you into adulthood. Animal studies have shown that a diet with too little methyl-donating folate or choline before or just after birth causes certain regions of the genome to be undermethylated for life. For adults too, a methyl-deficient diet leads to a decrease in DNA methylation, but the changes are reversible when methyl is added back to the diet. As shown in the figure, experiments in mice show just how important a mother's diet is in shaping the epigenome of her offspring. All mammals have a gene called agouti. When a mouse's agouti gene is completely unmethylated, its coat is yellow, and it is obese and prone to diabetes and cancer. When the agouti gene is methylated, as it is in normal mice, the coat color is brown, and the mouse has a low disease risk. Fat yellow mice and skinny brown mice are genetically identical. The fat yellow mice are different because they have an epigenetic mutation. When researchers fed pregnant yellow mice a methyl-rich diet, most of her pups were brown and stayed healthy for life. These results show that the environment in the womb influences adult health. In other words, our health is not only determined by what we eat but also by what our parents ate. As shown in the figure, chemicals that enter our bodies can also affect the epigenome. Bisphenol A, also known as BPA, is a compound used to make polycarbonate plastic. It is in many consumer products, including water bottles and tin cans. Controversial reports questioning the safety of BPA came out in 2008, prompting some manufacturers to stop using the chemical. In the laboratory, BPA appears to reduce methylation of the agouti gene. In the strain of mice that were studied, yellow mothers give birth to pups with a range of coat colors from yellow to brown. When mothers were fed BPA, their babies were more likely to be yellow and obese, like the one shown in the figure. However, when mothers were fed BPA along with methyl-rich foods, the offspring were more likely to be brown and healthy, like the one on the right. The maternal nutrient supplementation had counteracted the negative effects of exposure. So, a question may arise in your mind: if a pregnant mother's diet can affect the child's epigenetic outcome, can the dad's diet do the same? The answer is quite possible, according to scientists who delved into the well-kept historical records of annual harvests from a small Swedish community. As shown in the figure, these records showed that food availability between the ages of 9 and 12 for the paternal grandfather affected the lifespan of his grandchildren. But not in the way you might think. Shortage of food for the grandfather was associated with the extended lifespan of his grandchildren. Food abundance, on the other hand, was associated with a greatly shortened lifespan of the grandchildren. Early death was the result of either diabetes or heart disease. Could it be that during this critical period of development for the grandfather, epigenetic mechanisms are capturing nutritional information about the environment to pass on to the next generation? As we better understand the connections between diet and the epigenome, the opportunity arises for clinical applications. Just as mapping our gene variations gives us a window into our personalized medical needs, so might a profile of one's unique epigenome. Formed through a lifetime of experiences beginning in the womb, our epigenome may provide a wealth of information about how to eat better. Enter the future field of nutrigenomics, where nutritionists look at your methylation pattern and design a personalized nutrition plan. While we're not quite at that point yet, your doctor can already tell a lot about your disease risk by looking at your family health history. At last, you will be surprised to know that this epigenetics, or switching on or off of genes, can make a bee queen or a worker. To understand this, you have to closely look at the queen bee larvae. As shown in the figure, queens are raised in specially constructed cells called queen cups, which are filled with royal jelly. Royal jelly is a complex, protein-rich substance secreted from glands on the heads of worker bees. A larva destined to become a queen is fed large amounts of royal jelly inside a queen cup compartment. The larvae that develop into workers and queens are genetically identical. But because of her royal jelly diet, the queen will develop ovaries and a larger abdomen for egg laying, while the worker will be sterile. She'll also develop queenly behaviors, the instincts to kill rival queens, make communication sounds known as piping, and go on mating flights. The queen is fed only royal jelly for her entire life. In a series of experiments, scientists determined that royal jelly silences an essential gene DNMT3, which codes for an enzyme that silences a group of queen genes. When DNMT3 is turned on, the queen genes are epigenetically silenced, and the larvae develop into the default worker variety. But when royal jelly turns DNMT3 off, the queen genes jump into action, turning the larvae into queens. Thank you for joining me on this amazing journey of learning. We will meet again with new innovative topics. Meanwhile, don't forget to like, share, and subscribe to your own channel, ADP for Learning.